Good morning, good afternoon, everyone. Welcome to today's webinar. The webinar has just opened. I'm just going to give a couple of minutes to make sure everybody joins, and then we will start. All right, for those of you who has just joined, we are just giving a couple of minutes for everybody to join, and then we will start. All right, I'll give another 30 seconds, and then we will start. All right. So again, welcome to those that have since joined, since I gave the first greeting for today's webinar. We are talking today about Project Optimus from the FDA, webinar hosted by Bioforum and Cogitars, and we will look at how to tackle dose optimization according to the FDA's Project Optimus. Miguel, which I will introduce quickly next, will be the main speaker in this session, and I will be moderating. All right. Before we start, a few participation notes. You are all muted, so we cannot hear you, but there is an opportunity to ask questions. If you look on your right, there is a Q&A section. You can enter any question that you have as the webinar progresses in the Q&A session. We will have a session at the end of the webinar and answer all the questions as if time allows. Let me present myself first. I am Paul Milan. I'm heading up the biostatistics department at Bioforum. I've been in the industry since 2003. Most of my career I spent at a big CRO, and then I joined about 4 years ago the Bioforum group, heading up their biostatistics group. I've been a trial statistician for many years. I've managed teams of statisticians and statistical programmers, as well as sites, and at the moment enjoying my time here at Bioforum. And with that, I will introduce our main speaker today, Miguel. So if you can take it away.
All right. Hello, everybody. I'm Miguel. Just as a quick intro, even though I'm now a statistician, I was originally a medical doctor. And after that, because I was like mathematics and statistics, I ended up doing a PhD in stats. And since then, I've been working mostly in the exotic field of Bayesian statistics, specifically applied to clinical trials. And a few years ago, I joined Cogitars, which is a company originally founded in Germany. And I opened the UK branch of this company. So now we are between Germany and UK, working with people all over the world with interesting trials. And today we're going to talk a little bit about our experience with Project Optimus, since we work a lot in the oncology settings, especially in the early phase setting. And so now to the actual content. So why Project Optimus, and just a quick overview of what it is, and effectively why is this important, and why did the FDA ended up creating this initiative. So first, so the Project Optimus is a 2021 draft guidance from the FDA. It's effectively still a draft guidance sense, but it's effectively enforced by the FDA. And the primary goal, to read the text that comes in the actual guidance, is to reform the traditional approach to dose selection in oncology drug development, moving away from the maximum tolerated dose, or the MTD, paradigm to a more patient-centric model that emphasizes the identification of optimal doses, balancing efficacy and safety. And effectively, when we think about this, if we distance ourselves from people that work in the industry and that are used to the way things work, if I go and tell other statisticians how we do things in the industry, where, for example, we use things such as the 3+3 model, and I tell people, so we enroll 3 patients, we see if they have any adverse effects or adverse events, and then we enroll 3 more if we see at least one adverse event, and that's how we do the dose selection. People will be very astonished. So effectively, this type of model is not a model that should be working. It's been working fairly well with cytotoxic agents, as we'll see in a bit, but this low sample size, very simple way of addressing safety in dose selection based only on safety is effectively outdated. And one interesting statistic that I really like is that the 3+3 design has a targeting rate of 33%, meaning that in 2/3 of the trials, so of all the trials, in 2/3 of them, our MTD defined by the 3+3 is effectively wrong. It's not the real MTD, might be lower, might be higher. If the real MTD is lower, we usually tend to find that out when we enroll more patients, for example, in a phase 2 study. If the MTD is higher, meaning we might be in a suboptimal dose, both in terms of safety and efficacy. Definitely in terms of safety, so we could go higher. So effectively, it's not a fit-for-purpose methodology. And even though the industry seems to be switching a little bit, or going away from the 3+3 to other types of designs.